Hello and welcome back to educator.com. Today we're going to talk about gene regulation in prokaryotes. So as an overview, we'll first talk about transcriptional regulation and then we'll move into the regulation of translation. We're going to focus mainly on transcriptional regulation because that is most often where gene regulation occurs. So we'll talk about bacterial operons, and we'll focus on two of the most popular ones taught in many university classes, which is the lactose operon and the tryptophan operon. And then I'll talk to you about the virus bacteriophage lambda, and we'll talk about its lytic or lysogenic growth phases. And then finally, as I said, we'll talk about the regulation of translation. So, what is gene regulation? Well, it's the regulation of the amount of RNA that's made from each individual gene DNA sequence, okay? And so this is transcriptional regulation. Okay? This is often tied to the amount of protein that's produced from that gene. So usually if you decrease the amount of mRNA made, you will decrease the overall amount of protein that can be made. Now, regulatory proteins control our gene expression, and most of that regulation occurs at the step of the initiation of transcription. So that's important right here. Okay. So example, you could increase or decrease the binding ability of the RNA polymerase to its gene promoters. Okay. Now, regulation can also occur during the elongation or termination steps of transcription, as well as during translation. So let's jump right in. The bacterial operons. So what is an operon? All right, it is a functional unit of DNA that contains several genes all under the control of a single promoter. Now this is important. So several genes all under the control of a single promoter. Okay? So that means all genes will be transcribed in equal quantities. So the first operon we're going to talk about, the lac operon or the lactose operon. Okay, this is found in E. coli. The whole purpose is to metabolize lactose. Right? And this particular operon is regulated at the initiation step of transcription. So this is very common. There are 3 genes contained in this operon, LAC-Z, LAC-Y, and LAC-A. And it is what is called an inducible system. So meaning it is normally not being, there's little or no transcription, and it can be induced to have high transcription. So when there is high lactose concentration, there will be high transcription. If there's no lactose concentration, there will be no transcription. So let's draw this out. What does it look like? First of all, what does an operon look like? We're not even specifically going to talk about the lactose one yet. So what does an operon look like? Let's see. So if we check out our DNA, okay, 5 prime, 3 prime, okay, and we'll say we have 4 genes here, the A gene, the B gene, the C gene, and the D gene, okay, all under a single promoter, okay. Now, if we make the mRNA from that, okay, we have what's called a polycistronic gene, so it's multiple genes on the same mRNA. So we have our A, B, C, D genes, okay, and what we have here is that this is a fixed ratio of A to B to C to D because it's all under the control of the same gene promoter. Now, what this will make is then for example, what this will make is protein A. Protein B. Protein C. And protein D. Now, usually what makes an operon is that all of the genes under control of the single promoter act together in a certain way. So they act in concert. Okay, and an example of that would be turning, let's say we have substrates. Okay, our substrates, let's go purple. Okay, so protein A might work on substrate 1. Okay, so protein A might be responsible for turning substrate 1 into substrate 2. Okay, and then protein B would be responsible for turning substrate 2 into 3, protein C 3 into 4, and protein D 4 into 5. So what this entire operon does, okay, what the entire operon does is turn substrate 1 into the final product of whatever product 5 is. So this is the ultimate goal of this entire operon, and so it works in concert to do that. So back to our specifics, let's look at the lac operon again. So we have the 3 genes, LAC-Z. That's going to encode an enzyme named beta-galactosidase, and that is the enzyme that cleaves lactose, okay, which is a disaccharide, into glucose and galactose, 2 monosaccharides. We then have LAC-Y, which encodes lactose permease, and that's a membrane protein responsible for allowing lactose to get into the cell. Okay, so it allows you to get from the outside to the inside. And then we have LAC-A encoding an enzyme called galactoside O-acetyltransferase, and that's an enzyme involved in detoxification of the cell. All right. Now, also part of the lac operon, okay, is an upstream gene, so it's not under the same promoter, so it's not being made in equal quantities of LAC-Z, Y, and A, okay, in a normal thing. LAC-I is our upstream gene that encodes a repressor protein, okay, so a transcriptional repressor protein. And what this will do, it'll bind lactose if lactose is available. Otherwise, it'll bind the operator DNA sequence, and if this repressor binds the operator sequence, there's no transcription. Okay, so basically, if lactose is available, the repressor is binding lactose and not the DNA operator, and therefore the operon is active. Okay. So let's draw out this lac operon. So what's it look like? Let's look at the DNA. Okay, this is a way just to show that there's a lot of distance in the DNA. It doesn't necessarily have to be very close. And so this is the +1 site shown right here. And what we can make from that is our mRNA. And our mRNA will have our LAC-Z, our LAC-Y, and our LAC-A. Okay. Right here, LAC-I, that's our repressor. Cap, that's our cap binding sequence. P is the promoter. O is the operator. LAC-Z, Y, and A are our LAC operon. Okay. So, in times of high lactose, we'll just write this out here. We have high transcription. In times of no lactose, we have no transcription. Okay, so for the promoter to be active, okay, the cap protein must bind the cap DNA sequence, okay? And what for cap to bind, CAMP, cyclic AMP, needs to be bound to the cap protein, okay? If there is no CAMP, then we have no cap DNA binding, which means we have no transcription. Okay? Now, what's important is that, and I'll show you with the star, as your glucose concentration increases, CAMP concentration decreases, and that is going to lead to lower transcription. Well, where does glucose come into play? Well, remember, glucose is a breakdown product of lactose. Excuse me. So if I write this down here, lactose gets broken down by beta-galactosidase into glucose and galactose. Well, glucose, in fact, blocks an enzyme called adenylyl cyclase. And that enzyme is responsible for turning ATP into CAMP, CAMP. So, as glucose concentration goes up, we block more of the enzyme that turns CAMP. Okay, that turns ATP into CAMP. So therefore, as glucose concentration goes up, CAMP concentration goes down, and we see that CAMP needs to bind the cap protein for the cap protein to bind the DNA sequence, and the cap protein needs to bind the DNA sequence for transcription to occur. So it's all interrelated, all right? So this is all important. Now, we also know that LAC-I, the repressor protein that would be made, normally binds the operator sequence, okay, right here, which will turn off transcription. So we wouldn't get any mRNA, right? So this is all blocked off. We wouldn't do that. But if there's a bunch of lactose present, that LAC-I will actually bind the lactose. Okay, therefore freeing up the operator so that transcription can occur. Okay. Now, there are a couple caveats to this that I want to point out about the operator. So, if the DNA sequence is mutated, then the repressor LAC-I can't bind no matter what the lactose concentration in the cell is. So, if the operator sequence is mutated and LAC-I can't bind no matter what, this is what would be called constitutively active. Constitutively active or on, okay? Meaning high transcription. Okay, so that's if operator sequence is mutated. Okay, now another thing that can be altered is that if the LAC-I gene is mutated, then it doesn't matter how much lactose is present, okay, because the LAC-I gene won't be able to bind the operator sequence. So therefore, we have the same result as up here, constitutively on, leading to high transcription. So these obviously are not under normal circumstances. That's when we have a mutation. But this is just a couple other ways to see how this LAC operon can be regulated or actually lose regulation. All right, a second bacterial operon that we're going to talk about today is the tryptophan operon, or the TRIP operon. Okay, this is also found in E. coli. Its purpose is to produce tryptophan from charismatic acid. It is also regulated at the initiation step of transcription. This one contains 5 genes instead of 3. And it is derepressible, meaning that it is normally in a repressed state, and you can take that repression off. So if you have very low tryptophan concentration around, this will become active because you're going to want to produce tryptophan. Okay. So let's draw out the TRIP operon. So it has its own version of a repressor. Okay, that's upstream. We have the promoter, we have the operator. We have these regions called 1, 2, 3, and 4, which are part of an attenuation region. Then we jump into the operon, trip E, trip D, trip C, trip B, trip A. Okay. And then obviously more DNA. So what does our mRNA look like? Oh, sorry. The +1 side is right here. So we can get 2 different mRNAs. Now, this right here, remember, I said was called the attenuation region. This will affect whether we get a small mRNA or a large mRNA, and that will be due to the fact that one thing I want to point out is that all of these are inverted repeats. a thing about inverted repeats, they can bind to each other, so causing intramolecular base pairing. Another thing I want to point out is that sequence 1 contains 14 codons, 2 of which are tryptophan. Okay. Right? Okay, and so that's called the attenuation region. Okay. Attenuation region. So, first things first. We have our mRNA. Okay, here's our 5 prime. All right, we have all the way through. All right, we have trip E, D, trip C, trip B, trip A. Okay? All right. Now, this is when we will get this transcript. So this is called a variant, an mRNA variant. We get this one when we have low tryptophan concentration. Okay? When we have low tryptophan concentration, we get this. And the reason being, what happens is that we actually get this attenuation region looking like this. So we have 1, 2, 3, 4. Okay? And what this is, we call it a tennis racket or whatever you may want to call it. But we have intramolecular base pairing between region 2 and region 3 because we have low tryptophan, so we're having this intramolecular base pairing between 2 and 3, right? Now, this leads to a row-dependent termination, okay? And this mRNA is over 6,000 bases long, and we know it's polycistronic because it has all these genes. Okay. Now, there's the other variant that can be made when we have high tryptophan. Okay. So it will end here, okay? And it's actually only 139 base pairs long, or bases long. Okay, so this right here is the plus 139. Right there is the plus 161 spot. This will end up making a 3-4 hairpin. Oh, here, I forgot to write out here. So what we have is then EDCBA. Okay. And this is what we call the attenuated version. And this would undergo row independent termination. This right here, this type of hairpin between the 3 and 4 actually causes the mRNA to pull out of the RNA polymerase, so it pulls it off of the DNA, resulting in an attenuated or shortened mRNA, whereas the one up here, the hairpin between the 2 and 3 region, is rho dependent, so it won't pull out until the rho factor comes after recognizing the stop sequence at the end of the mRNA. So that's the difference here. So when you have low trip, you have a 2-3 hairpin, and that allows you to transcribe all the way through to the end of the sequence. When you have high tryptophan concentration, you have a 3-4 hairpin, so up here in these attenuation regions, which causes rho-independent release of that mRNA and an attenuated only 139 base mRNA. Okay. Now, one thing is important to mention is that when we have this high tryptophan concentration, the repressor will bind the operator sequence, leading to no transcription. Up here, when we have low tryptophan concentration, the repressor does not bind the operator sequence, which leads to transcription. Okay. And one thing to point out, remember, I said in this one sequence right here of the attenuation region, it contains 2 tryptophan codons. If you have absolutely 0 tryptophan, the ribosome will stall at that attenuation sequence 1, okay, because it has 2 trip codons. Okay, and if you can't put in trip right there, it can't continue, the ribosome can't continue on synthesizing new protein, okay? Now, this attenuation is a way to regulate gene transcription about 8 to 10-fold. You can do repression using that repressor protein, gives you regulation about 70-fold, and attenuation and regulation together can give you overall gene regulation of anywhere from around 600-fold. So these are just different ways between the lac operon and the trip operon of regulating gene expression, and in 2 completely different ways, even though they're both regulating at the same part of transcription, which is the initiation. Okay. So let's move away from our bacterial operons and let's switch gears to looking at how a virus works through its life cycle. So we have bacteriophage lambda, okay, and that is a virus, and it infects the bacterium E. coli. It has what's called a temperate life cycle. It can either be lysogenic, which means its DNA genome, okay, here's an example of a bacteriophage. It will latch onto an E. coli cell. Okay, this is E. coli. Okay, with E. coli's chromosome, right? We'll just say that. The lambdaphage will shoot or inject its DNA into the E. coli, and it will make a circularized genome inside the E. coli. So this is the lambda DNA. Okay, so there are 2 things that can be done. Either this DNA can integrate into the host genome, okay, so becoming one with the E. coli DNA, and it can just stay there dormant or silent, kind of just outside of view. Or the lambdaphage can just replicate its genome separately over and over and over again, hijacking the E. coli cell machinery, making many new viruses, and it will just lyse the bacterial cell, and which in fact kills it, and release more viruses to the environment, in which case those can do the same thing. They can be infecting more E. coli, and then based on what they want to do, they can either integrate into the genome for lysogeny, or they can just hijack the cell machinery and just use it to produce more and more viruses and lyse the E. coli. Okay, so this is how the bacteriophage can effectively regulate its transcription to do one or the other. So let's draw it out. It's a complex process. I'm only going to draw out a simple part of it, but we're going to look here. Okay, so this would be the DNA of lambdaphage. So this is the lambda DNA. There's much more to it, but we're just going to focus on this. Okay, so right here, I'm just going to write it out and then I'll explain it. So this is, remember, this is DNA. So the CRO protein that can be made from the CRO DNA sequence will bind at this spot. When CRO binds here, okay, this, by the way, sorry, PL, PRM, PR, PRE, those are all promoters, okay, and that's why they have the directionality. So is which way they will promote transcription. So we have 3 of them pointing in the leftward direction, just PR pointing in the right direction. So if CRO binds this region, this OR3 region, then that will block the PRM promoter. This will lead to PR and PL, okay, so PR right here, and PL, they will be the promoters in charge of transcription. This will lead to lysis of the bacterial cell. Now, C1, which can be made, a protein that can be made from this DNA sequence, will bind, let's draw in a different color, to this sequence. Okay. If C1 binds, the PR promoter is blocked. And we will have PRM being the one that is transcribing. Okay. This will lead to lysogeny. So entering into the bacterial DNA. Okay. Now, why would this occur? Okay, so this is important. So I will write it with a star. C1 concentration must be kept high to stay in lysogenic phase. Okay. And how it does that is we have the pre-promoter helping out in this. Helps keep CI concentration high. Okay? Because the C1, not CI, C1 is under control of this pre-promoter. All right? So, basically, if the pre-promoter, okay, is active, C1 is produced quite highly. If C1 is produced, we have it binding to this spot, blocking the PR promoter, which keeps this PRM promoter active, and it allows you to stay hidden inside the E. coli cell. If you don't have C1 being made very high, we have CRO being made. So if you're not going in the leftward direction, okay, the leftward direction is producing C1. The rightward direction, okay, the PR and PL, okay, the rightward is making CRO. Okay, so if you're going rightward, we have a lot of CRO around, CRO protein. That's going to bind this one, blocking the PRM. Okay, the PRM and the PRE are going to help with a C1. Therefore, this is going to end up going through a lysis phase. So it's going to end up just hijacking the cell machinery, killing the bacteria, and exiting out. So if you don't regulate transcription and translation properly, then you're not going to be able to contain whether you're going through lysogeny or lysis. And now, other than transcriptional regulation, DNA damage can play into this. So, for example, if we have bacterial DNA damage, that can actually shift you from lysogeny to the lytic phase. Okay, and how does it do that? Well, when you have damage, you have DNA gaps occurring. DNA gaps are bound by Rec-A, binding the single-stranded DNA. Rec-A gets activated. To Rec-A star. And then Rec-A star causes autocleavage of the C1 protein, which leads to lysis. So not just transcriptional regulation, but actually DNA damage can affect whether you have the lytic life cycle or the lysogenic life cycle of the bacteriophage. So this is just separate from an operon, but it is a very involved DNA sequence that goes through transcriptional regulation, and depending on which protein is found in higher quantity, C1 or CRO will tell you whether you are going through lysogenic phase for C1 or lysis phase for CRO protein. All right. Now, I briefly want to go over the fact that we can be regulated at translation, and what that is normally looking like is that you bind the RNA by proteins near the site that the ribosome would normally bind. So therefore, the ribosome can't even bind the mRNA. So this doesn't allow the 30S ribosomal subunit to bind RNA. Okay. Another way that you can regulate translation is if you have intramolecular base pairing of mRNA. So the mRNA itself could be hiding its normal ribosome binding site. Maybe it's right here. But because it's base paired to itself, the ribosome can't bind the mRNA. And then there's also post-transcriptional regulation of rRNA folding via translational repression of the ribosomal protein synthesis. So basically, if you have rRNAs not folded correctly, they can't interact properly with the proteins to make the nucleoprotein that we know as the ribosome. I'm not going to go into any one of these specifically, but these are all options and possibilities how you can regulate translation. So for the last slide, I just want to give you an example of something that is actually a pretty cool phenomenon. So we have this piece of nucleic acid called the SSRA RNA. And this is a really important RNA found in prokaryotes that can rescue ribosomes that are translating broken mRNAs. Now, or an mRNA that lacks a stop codon. Now, this is really important because if we have a ribosome, okay, and it is translating the mRNA. Okay, this is our eukaryote or prokaryotic, so this is a 50S and a 30S to make the 70S ribosome. So if we have an mRNA that's stuck in a ribosome, okay, so it's a broken mRNA, it gets stuck in the ribosome, that ribosome will continue to try to translate it, but it's basically not happening, okay? So basically what this does is it takes that ribosome out of commission, right? So until that mRNA can be released, the ribosome is no longer useful for translating any functional proteins. Same thing that if an mRNA doesn't have a stop codon, that can be problematic. So our ribosomes need to be fully functional. So if we are translating a broken mRNA, the ribosome is just basically going to stall there and wait until something can be fixed. Well, since that normally doesn't happen, we have this special RNA coming in, and it'll rescue that. And it is what is called a tmRNA. So it's part tRNA, part mRNA, and it can be charged with an alanine amino acid. Okay? Now, what happens is if it's stalled right here, this tmRNA can come into the A site. Okay? Okay, and it can be charged with an alanine. Okay, it can come into the A site, allow translocation. So go to that P site, right? So the ribosome will be going that way, which means that this will go to the P site. And what it does is translocation is able to occur, and then it's followed by 10 codons and a stop codon. So the protein that's coming out, right? So the protein that's coming out, maybe it was stalled right there, but then you have this tag that is added. We'll make it in green. So it's 10, okay, and then a stop codon, right? Now this is a very important piece. This acts as a tag that directs the destruction of the mRNA by the cellular proteases. So this is, that directs the overall protein that was produced, not the mRNA. Okay, so that protein gets sent to the proteasome and it gets destroyed. So this is really important. So it allows the mRNA to be released from the ribosome. Now the ribosome can pick up on another mRNA and be functional, making a functional protein on that, as well as this messed up protein that was made from a broken mRNA or an mRNA without a stop codon, can then be just degraded and thrown away so that it doesn't fold into a protein and maybe become a protein that screws everything up and maybe has a function that was unintended. Okay, so this is a pretty cool RNA feature and a protective feature that is found for translational regulation in prokaryotes. So I hope you enjoyed the lecture today. Thank you for being here at educator.com, and I hope to see you again.